Hi, my name is Jerry Palmer. I'm part of the Qubit team that designed the software for the gas switching apparatus. There are three other videos in addition to this overview video, and these other videos discuss various aspects of the switcher, some of the concepts of switching, the features of the Logger Pro software which runs the switcher, and finally there's a assembly video produced by Monika Kuzma in which she discusses how the apparatus is assembled. A switcher, or another name for it, multiplexer, is an instrument that allows you to analyze several samples with a single set of sensors. In our business, we're usually dealing with respiration gases such as CO2, oxygen, and perhaps methane, and we're analyzing the concentration of these gases from respiration samples. The switcher is often connected to a Q box which contains the gas analyzers, pumps, flow monitors, etc. Qubit makes several different Q boxes which can be coupled to the switcher. The RP1LP is designed to measure respiration from small animal and even insects. An RP2LP for larger animal respiration, a CO650 for plant photosynthesis, and an SR1LP for studying respiration from soil bacteria. Here I show the overall design. The switcher is coupled to a Q box. The Q box pumps in a carrier gas, passes it onto the switcher. The software selects a particular sample for the gas to be passed back to the Q box, where flow is measured and the concentration of CO2 and O2 is measured before the gas is vented to atmosphere. The carrier gas can be one of several types. It could be tank air, commercial tank air, nitrogen, or the cheapest method is to use air scrubbed of CO2 with soda lime. The air should also be dried so that the water vapor doesn't dilute the gases of interest. If you're not interested in having CO2 free carrier gas, I would suggest you simply use outside air that's been dried, since the CO2 in outside air is quite constant at about 400 ppm. I'll now discuss the different configurations the switcher can be assembled in. There are really two: an open flow configuration and a stop flow configuration. The open flow configuration is designed to look at large respiration rates from larger animals. It has continuous flow through the chambers. In other words, the specimens always have gas passing by them. The selected one, the gas is passed back for analysis. The non-selected specimens, the gas is vented to atmosphere. The data of interest in open flow is the differential. In other words, the difference between the gas concentration in the selected sample and the reference. Channel 1 is always reserved for the reference. Usually the reference means simply an empty chamber so that you can measure the background carrier gas. So this difference measurement allows you to calculate VCO2, the CO2 production, VO2, the oxygen consumption, and RQ, which is the respiratory coefficient and is simply the VCO2 divided by VO2. RQ is quite useful in studying metabolism, the type of food being metabolized. The open flow method has higher time resolution but lower sensitivity than the stop flow, which I'll discuss briefly. So now more of the details of the setup. The carrier gas is pumped into a regulator. The purpose of the regulator is to provide a constant pressure so that when you control the flow through a particular channel using the valve on the flow meter, you don't affect the flows in the other channels because if you have constant back pressure. When the gas comes out of the regulator, it's split into the various channels, passes into the flow meter, or as they're called, rotometers. They each have a valve used to control the flow, and they provide a visual measure of the flow as well. When the gas comes out of the flow meter, it passes into the chamber. The selected chamber, the gas is directed from the switcher back to the Q box for analysis. The non-selected chambers, the gas is vented to atmosphere. In order for these vents to be open, ensure that the bulkheads have these small brass adapters screwed onto them. The selection of the chamber is controlled from Logger Pro, the software, through what's called a DCU, Digital Control Unit. This digital control unit outputs three signals, D1, D2, and D3, which can be used as bits, and 3 bits will let you count up to 8. So these are passed to the microprocessor within the switcher, the G244 switcher, and the microprocessor uses those bits to count a particular channel that's to be selected for analysis. I'll now discuss stop flow configuration. As I said earlier, it's designed for very small respiration rates, such as that from insects and larvae. You can actually measure down to just a few small insects, such as Drosophila or mosquitoes. In this method, you actually halt or stop the flow through the chamber for a certain length of time and let the gases build up in the case of CO2 or deplete in the case of oxygen. Then you resume the flow and sweep these gases, these built up gases, out for analysis. So what you end up seeing is a peak in the case of CO2 or a trough in the case of O2. The data of interest is the area of these peaks, and in combination with the stop time, you can calculate VCO2, VO2, and hence RQ. The stop flow configuration has a much higher sensitivity but a lower time resolution than open flow. It has high sensitivity because you can leave the chamber stop for as long as you want to let the gases build up. This of course lowers the time resolution because you can't analyze another sample until that stop time is finished. So now I'll discuss the details of the stop flow configuration. It's very similar to the open flow configuration. The only difference is each chamber has a pair of 3-way solenoids around it to allow the gases to be bypassed around the chamber. So when these solenoids, shown here in this picture, are non-activated, the gas passes around the chamber. The chamber is stopped, in other words. When the solenoid is activated in the selected chamber, the gas actually passes through the chamber to sweep the stop peaks out to the analyzer for analysis. So just as in the open flow method, the carrier gas is pumped through the regulator, split into the various chambers. The flow is controlled by the valve on the flow meter. This is the flow meter, or so-called rotometers. Each one has a valve at the bottom to control the flow, and it also has a ball in a column where you can visualize the flow. So these gases are coming into the back of the flow meter. They come out of the front of the flow meter to the solenoid in the case of stop flow. If you're doing open flow, these solenoids would be removed. Okay. The state of the solenoid determines whether the gas is bypassed or pushed through the chamber, depending upon whether they're activated or not. Non-activated solenoids, the gas is bypassed. Activated solenoids, the gases pass through. So the gas coming out of the flow meter can either be shorted back to the switcher. This is shown as a 4-channel switcher. Back to the input. The non-selected ones are vented through the open vent ports. The selected chamber, its gas comes out the analyzer out port and is passed back to the Q box for analysis. In the case of stop flow, you're likely to be using these types of chambers. They're glass chamber with a movable plunger, so that you can vary the volume from anywhere about a half a mil to about 12 to 13 mils. So it's designed to be used for fairly small insects right up to larger specimens. In the case of open flow, where these solenoids are removed, the chambers will possibly be larger chambers, since the specimens may be larger. This is a photo of a Q box. The Q box consists of various modules such as pumps, flow monitors, gas analyzers, CO2 and O2, and these modules are standardized in size so that they can be interchanged, and you can easily change one Q box into another by simply rearranging the modules. So in other words, one type of experiment can be easily changed to another. This is quite useful for teaching labs. The modules fit into a tray. They're plumbed. The gas is dried before it is analyzed by the gas analyzers. The calibration is performed on the gas analyzers after the tray has been placed back into the weatherproof suitcase. Once it's all calibrated in the suitcase, this suitcase can be taken to the field and operate on battery if